Hi there. Welcome to Mushroom Hour. Today on Mushroom Hour, we're fortunate to be joined by Alison Pollack. Alison is a photographer specializing in making large the diminutive dwellers of the forest, Myxomycetes, commonly referred to as slime molds, as well as other tiny fungi. Her passion is finding, photographing, identifying, and sharing these miniature worlds to bring awareness to the fascinating organisms that exist right at our feet in the forest, but are largely hidden to the naked eye. Alison has always had a casual interest in photography, but when she retired from the field of air quality consulting, she upgraded to a DSLR and began shooting landscapes at home and while traveling, hiking with her husband throughout the world. While hiking locally with her new DSLR, she began discovering the diversity of mushrooms that fruit in the Bay Area's winter rains. It wasn't until she found and photographed her first slime mold, however, that she got serious about mushroom and myxo photography. Alison's photographs have now been featured in numerous publications, including Colossal, Bored Panda, My Modern Met, the German and National Geographic magazine Geo, Der Spiegel, and the Sunday New York Times magazine. Known as Marin mushrooms on Instagram, her widely shared posts have inspired people to slow down on their hikes and to search for these tiny life forms that she loves. Alison, thank you so much for coming on the show.
You're welcome. Thank you for inviting me.
I'm sure I speak for anyone who's found you on Instagram or on Facebook that we've just been blown away by the captures that you get of these tiny, tiny organisms, Myxomycetes and tiny fungi. There's something amazing about making those little organisms big that you do so well. And I want to talk all about that journey for you as a photographer to develop a relationship with these organisms and learn how to photograph them and capture them. But I am curious how you got started, how you caught the bug to search for Myxomycetes or slime molds.
Not the bug. As you said, I've always carried a camera with me. And I did start, once I got a good camera, I did indeed start losing my hiking group. I would take some pictures and then I would run to catch up. But it was actually on just a hike with a friend with only my cell phone that I saw in a redwood forest decomposing logs, something yellow, bright yellow, which caught my eye. So I went and looked at it and I had no idea what it was. It looked like little strings of tiny, tiny peppers. And I'd never seen anything like it before. And I was mesmerized. So, of course, I took a photo with my cell phone and I went home and I looked it up on Google and I found it pretty quickly. And I found that it was something called slime mold, which I'd never heard of, Myxomycete. It was Leocarpus fragilis, by the way. That was the very first one. And I ended up reading until about 4 a.m. because I was fascinated, reading and looking at photos. I was fascinated by what I saw and I just couldn't stop reading it. I was instantly smitten. And I went and said, okay, I'm going to find more of these and photograph these. And I bought my first macro lens and headed back into the woods.
And before this interview, you qualified that you're not a biologist and you didn't want to dive too much into all the specifics of this life form. But I am curious what about them or what in that 4 a.m. kind of feverish reading session really captured your imagination?
I would say 2 things. One, the colors, the range of colors in these tiny, tiny, tiny life forms is amazing. Every color of the rainbow you will find in slime molds.
Wow.
So that's one thing. I tend to like bright colors. So a lot of them when they're young are very brightly colored. And the other was their life cycle. So they will change in color, shape, form, structure, everything in a matter of hours or days or maybe a couple of weeks. They just change amazingly. And I'd never seen anything like that before.
That's incredible to think of something that's able to transform. I mean, we all transform as biological organisms, but something that can transform so quickly and so vividly, with color and shape. They are fascinating. And your photos are really what kind of introduced me to this world. And I kind of share the same fascination you just described there because of color and the way they change. When you're out, I'm really curious because obviously for me, I'm very much a forager. I go out in the woods looking for edible mushrooms, looking for edible wild plants. But in your case, you're not really foraging. You're not looking for edibles. You're searching. You're seeking. So what is that process like for you of searching for Myxomycetes or slime molds? And then any tricks that you've learned to spot these little organisms?
Do you have a couple of hours?
Yes, multiple podcasts. This is actually going to be a mini-series.
Well, I laugh at foraging because the vast majority of people who are into mushrooms for
Right.
foraging for eating. And I do love mushrooms and I do love wild mushrooms, and I'm happy to let other people do the foraging. But for slime molds, I would actually let's start with something of the name slime mold, okay? So it's a pretty unfortunate term in my opinion, because those two words, slime and mold, both tend to conjure kind of ick among people when they hear them.
Right.
And yet, if I say Myxomycete, which is the botanical name, it sounds a little heady or a little snooty. And I think that slime molds need a marketing person. We need a better name. So I
Yes.
tend to refer to them as Myxos because that sounds kind of cute. It's not as fancy as Myxomycetes.
It's catchy.
Yeah, so everybody refers to them as slime molds, but it's, when you first hear that term, really, everybody says, ew.
Right.
Okay, so back to searching for slime. See, I did it myself. Back to searching for Myxos. So people ask me, can I go on a slime mold fungi walk with you, or and to photograph? And I say I either walk or I photograph. I don't do both. Because when I'm going into the woods looking for my subjects, I basically find a place where there's a nice wet decomposing log or a nice leaf litter, and I sit on the ground or on my hands and knees, and I take a magnifying glass. My favorite tool in my camera bag is a small magnifying glass that has LED lights on it. It's a 10x magnifying glass. And between that and some strong reading glasses, because my eyes aren't great, I will scan a log very slowly, very carefully with the magnifying glass and look for little tiny life forms. Now, once your eyes are trained to look for them, they're fairly easy to spot. When they're young, they're brightly colored, typically. So they're much easier to spot. When they're more mature and they're dried out and the spores are ready to be released, they tend to be darker colors, and then they're a lot trickier to spot, especially if they're against dark wood. That's why you have to go very slowly on a log. They also like to hide in crevices in logs. So if you have a decomposing log that's got a big crack in it, look inside that crack. They're very commonly in there too. The very first time I did this, I sat down at a pretty good sized log. Maybe it was 3-4 feet long or something. And I sat there for 4 hours in one spot on this one decomposing log. And I kept looking in little cracks and crevices. And in this one log, I must have found 10 species of Myxos and tiny Ascos. So I was there for 4 hours. And since then, that's not uncommon for me. I can easily sit for hours at a single log. A lot of people ask me, I keep looking and I can't find them. And most of the time it's because they're not looking closely enough. They think they're, a few inches tall or a millimeter, maybe a centimeter tall, but most of them are 1 to 3 millimeters tall. They're really tiny.
Tiny.
If I sit in the wet forest and I just stay there and I look around me, within 2 feet or so around me, I'm virtually guaranteed to find something, whether it's slime molds or tiny Ascos, virtually guaranteed.
And that was my question. Is there specific habitat you need to look for? Is there a seasonality? Do they only show up at certain times of the year?
They need wet. They need wet, humid environments. So in the Bay Area, they show up for our winter rains, which are getting shorter and shorter period, unfortunately.
Right.
So in the Bay Area, we need a good 1 or 2 good soaking rains and then some consistent rain for them to flourish. So they show up where I look, they're in forests, mostly in forests. You can actually find slime molds on people's lawns if they're watered, but mostly in wet, really nice wet forests where there's decomposing matter. So you need decomposing wood, fallen branches, or leaf litter. If you poke around in wet, damp leaf litter, you'll probably find Myxos as well, although it's harder to find them in leaf litter.
But it sounds like once you go to the forest and there's those right wet conditions, you really just have to hone in on one of these areas where there's some kind of decomposing wood and, more than likely find something. It's all just about looking hard enough or looking closely enough.
Yeah, I wouldn't say that every decomposing log has some. I wouldn't say that. But if you're in a fairly small area with a bunch of decomposing logs, you're pretty likely to find something if the conditions are right. Yeah.
Now, we did talk about the seasonality. So obviously there's probably times of the year it's too dry. Clearly, we're thinking of this summer where we just had heat wave after heat wave. Too dry to find Myxos. So you've dabbled in actually growing Myxos. Tell us a little about that. How have you tried to cultivate Myxos for when they're not in season?
Yeah, so this summer has driven me crazy because normally I would travel someplace. To be without an actual fresh Myxo or Asco in the woods since March for me has been just driving me crazy. I'm just counting the days and waiting until we get our rains. So I did try somewhat successfully to grow them, and it's actually fairly straightforward. There's a technique that was accidentally discovered in the 30s, I think, and it's referred to as moist chamber studies. And scientists use them, study slime molds in petri dishes. You can do it in a petri dish, you can do it in a deli takeout container, you can do it in a plate covered with saran wrap.
Yeah.
So you basically get a piece of bark. You can either take live bark from a tree, just a small piece of live bark, or just the dead outer layer of the bark, not the inner live part of the tree. Or I typically just take some decomposing twigs or branches or bits of decomposing wood from the forest. In the summer, it'll be dry. So I take it home, put it in a container, and let it soak completely in water, 8 hours overnight, something like that. So the wood, the bark, whatever it is completely wet. Pour out the water, and then put some damp paper towels, damp, but not too wet. You don't want water running. Damp paper towels at the bottom of your container, put your wood on top of that, and close the container. Well, here's the trick, which I haven't actually figured out. It requires the right amount of humidity, but not so much that you get mold. And in the summer, with the heat waves that we've been having, mold has been a huge problem. So I'm still experimenting with how much airflow, how much to let the top off, how much to cut holes in the sides. I do get mold fairly often, but I have managed to grow some Myxos and some tiny Ascos, and that's pretty exciting, especially because one of them was one I'd never seen in the woods before. It's called Arceria pomiformis, and it turns out to have this just gorgeous internal structure. And one of my favorite photos, which was taken with a microscope lens, was after I blew out the spores. You can see the threads that hold the spores together. It looks like incandescent light bulb filaments. It's really amazing. It's very cool.
Now, were these Ascos and Myxos that were already present on that piece of wood? You didn't have to introduce anything to that wood.
Right.
You just
I'm
had to
not
make the
doing any
environment.
inoculation or anything like that. So as you know, spores are all over the place. We're breathing in spores all the time. And that's certainly the case for anything you bring home from the woods as well.
Oh, very good. Well, now I want to talk about how you're actually capturing these. Step one, you go out in the woods, the right time of year, the right environment, you find them. But then to capture them to the level that you do, where, they're basically works of art, these pictures of the Myxos made large are just stunning. So I know when you first got started, you were a more casual photographer. How have you grown as a photographer since this obsession started?
The key word is obsessed. Yes, I have become obsessed. Right. Totally obsessed. For example, I used to read, easily a novel a week. I haven't read a novel since I became obsessed.
Myxos dominate all free hours of the day.
Well, I'm learning the craft of photography. Doing the magnification at the levels that I do is actually very technical. So it's taken me quite a bit of time and practice. So I have really become what I would certainly call an extreme macro photographer, and that's my specialty. Right. And I love learning more and more about it, and I'm always learning. I mean, I'm on my next stage now, which is to build a rig. I bought a microscope lens a few months ago, and it is incredibly sensitive to any vibration. So my husband jokes that when I'm using that lens at home is where I do it. When I'm using that lens at home, he can't move. He can't even walk around the house. He can't move. Most of my photography to date has been on a tripod, but even the tripod will pick up vibration. For example, when I'm home, I shoot my subjects in my studio, which I jokingly refer to as my studio, which is my kitchen counter. And the tripod is on the kitchen floor, which will pick up vibrations from the refrigerator motor, for example. I'm in the process of designing and building a rig for the macro, which is basically a very, very, very heavy base stand. And I have a series of rails to mount the camera with the lenses, and everything is very, very, very stabilized. So there's no movement, and that will allow me to get really sharper photos. I mean, my photos are sharp, but I want them even sharper. Yeah.
So are you taking any pictures out in the field, or are you taking most of the specimens home?
Well, now, of course, I'm only home because there's nothing in the field. But during the season, I much prefer to take specimens in the field. Far, far, far prefer that. I prefer the natural setting and I prefer the natural light. I try not to use any artificial light in the field. If I'm way under a log, then I have to. Then I use an LED light panel or something like that.
Right.
But I really prefer to use natural light. It's just softer, it's prettier, it's much nicer. Then if something, and I only got the microscope lens at the very end of last winter. So I
Okay.
imagine this winter I will probably do maybe half in the field and half at home because I'm so fascinated by what you can see with the microscope lens.
Yeah, that's going to be a whole new era of Myxo pictures, or the microscopic.
Yeah. Myxos are really cool in another sense, which is you take a mushroom from the woods, okay, and you take it home, it will shrivel up. It won't retain its beauty. You can use it for microscopic analysis, but it won't retain the form and the shape that you see in the woods. It will just dry up. But Myxos, they will dry a little bit, and then they will retain that dry shape. So there's a photo, I guess I haven't posted it yet, of something I found in my garage from a year ago, and it still looks fresh.
Okay, so they actually hold a certain form. They don't just
They
totally
do. They
shrivel
do.
or
So I'm
decay.
tempted. It's not a great picture, but I'm tempted to post that picture just to say, hey, another amazing thing about
Super
slimes.
cool. Yeah.
They last forever. That photo is also really, it's not a great technical photo, but it's also really cool because at the base, the Myxo is maybe 1.5 millimeters tall. But
Okay.
with a microscope lens, at the base, I can see Zygomycetes and all kinds of other tiny little things. So that's what I'm really excited about this winter, that I can go into the forest and take the slime molds to pick them home. But, the Zygomycetes in these really little, ridiculously tiny things, there's no way you can even see with the little magnifying glass. So
Right.
I'll take these home to photograph the Myxos, but I am sure that at the feet of these Myxos, I will many, many, many times find some ridiculously tiny fungi.
So you're showing us
Yeah.
the microscopic forest within the forest.
Yes. Yeah,
That's
it's really
fantastic.
cool.
When you're out in the field, your tool of choice then is the DSLR. Obviously, we're not talking about an iPhone with, a little macro lens stuck on it. What kind of lens are you using on the DSLR to capture the pictures?
Yeah. So I have 2 cameras. I just bought a second camera about 2 weeks ago, which I can talk about. But
Yeah.
until now, all of my pictures have been taken with a Sony full frame camera, Sony A7R2. But, a lot of camera and lens combinations can take these pictures in the woods. So
Okay.
it's not that important for me to say specifically. The main thing is that I have a macro lens. And what a macro lens is technically means that you're capturing something life size. So the sensor on a camera, on a full frame camera, is 24 millimeters tall and 36 millimeters wide. A macro lens, which life size means that what you're photographing, it will fill the frame if it's 36 millimeters wide and 24 millimeters tall. It will fill the frame.
Right.
That's a lot of Myxos because they tend to be, Stemonitis might be 6, 7, 8 millimeters tall, but most of them are 1 to 2 millimeters tall. Well, I have a 24 millimeters of height, so you can get a wide scene.
Right.
So that's actually almost too much for what I want to do.
Yeah.
So a second lens that I take out with me in the woods, it's not actually a lens, it's a diopter, an add-on to a lens. It's a great little tool. This one I do want to give a plug to, and I'll put it on at the end of the note. It's called Raynox, Raynox. It's a diopter that will basically more than double the magnification of your lens. You add it onto the end of your macro lens, and it's fairly inexpensive, and it's amazing quality given the price. And so in the field, most of my pictures are done with my macro lens with the Raynox attached to it. And that gets me to a magnification of about 2.5 times life size.
Yeah.
Okay. So that means instead of 24 by 36 millimeters, you just divide that by 2.5. So we're getting something like 10 millimeters by 15 millimeters, something like that.
For these truly small organisms, you need that level of magnification to really capture them. And are they in situ? I mean, are you taking the photograph right there on the log? The reason I ask is I know sometimes with mushroom photographers, now obviously we're talking much larger organisms, but they'll take the mushroom, place them somewhere else, do some kind of uplighting. But are you doing most of your photographs actually just in situ with just the natural light?
In the field, I would say the vast majority are in situ with the natural light. Sometimes if it's so dark and so deep, and if I can move it somehow, either cut out a tiny little piece of it or move the log, I'll raise it up so that I get more light in the background.
Yeah.
The other thing is that I always want to have the viewer focus on just the Myxo and
Right.
not the background. So
Right.
I'm always looking for, when I set up the composition, I'm looking for a background that's distant. If the background is very close to the Myxo, then it's going to appear in the photograph. I want it to be distant. So I want to talk about the most important thing I do for photography is a particular technique that I do called focus stacking. Myxos are very tiny. The tiny ascos that I photograph are very, very tiny. When you use a macro lens with, say, 2.5 multiplication, which is pretty much what I'm doing in the field, your depth of field is ridiculously small. And depth of field is the amount of your subject that's in focus. So the depth of field is very, very small. And so if you just take a single photograph, you'll get, and let's pretend it's a mushroom, you'll get a part of the mushroom in focus, but not the whole mushroom.
Okay. That makes
Okay.
sense.
So what you do in focus stacking is you take a series of photos from the front to the back of the mushroom where you change the focus spot a tiny, tiny bit each time. Okay. So each photo you take is further back on the mushroom and it's a little bit different part of the mushroom in focus. And then you have a series of, say, 10 or 20 or 30 photos where each one has something different that's in focus. And there's software that then takes those photos and takes the pixels, literally, that are in focus in each photo and combines them, composites them into a single photograph. The first time I did that compositing on my computer and the photo showed up on my screen, I literally said, wow, and jumped up out of my chair. I just thought it was magic. It is still magic. So when I'm in the field, I do that focus stacking, and I'll typically take, I would say, between maybe 20 and 60 photographs in the field.
How long does each one take when you're having to focus on just little, less than millimeter segments of each organism, each Myxo?
Yeah, it's definitely way less than millimeter segments.
Right.
And so it takes some practice. And what I do, so I have a remote control for the camera. So again, you never want to press the shutter. The act of pressing the shutter button creates vibration and creates blur in the photograph. So I always use a remote. And you have to have a very sturdy tripod. You have to have the same position, the same angle of the subject at every photo. So I set up my composition, which takes me actually a long time. So let me back up. Not so much with Myxos because they're harder, but with tiny fungi, I will clean. A friend of mine calls it custodial duties. I will clean my subjects. Whatever's around it, if there's little specks of dirt or little tiny, not even twigs, but little tiny bits of wood, I have tiny tweezers that I take with me. I clean all that. I have paintbrushes. If it's a tiny mushroom, I will clean the mushroom with very, very delicate paintbrushes. I can easily spend half an hour, 45 minutes just cleaning the area for the photograph. So before I even take my first shot. So I'll clean my subject as well as I can, and I will clean the area that's going to be in the composition.
Right.
Then I take a series of photographs to check the lighting, to do the adjustments. I don't use any automatic settings on my camera. Everything is manual. So I take a series of photographs to get the lighting right. If I have to use artificial lighting, I check that. Occasionally I use flash, but again, I really prefer not to do that. So once it's all set up and I know everything's set, I focus in on the very front of the scene, whatever that is, the first thing I want to be in focus. Actually, the truth is I usually go a little more towards myself for that. In case I've missed something, you can always throw out photos, but you can't go back into the woods and get them later. Right. So I've learned the hard way to always take extra shots. I focus on what I think is the front, and then I pull even more towards myself. And I take my first shot. I move the focus ring a tiny, tiny bit on the camera, just with my fingers. I just rotate the focus ring on my camera a tiny bit. Let the camera settle. You're touching the camera. So I give it a couple of seconds to settle. I take another shot, always using a remote. Rotate the focus ring again a tiny bit with my hand. Let it settle. Take the shot, etc. So if I'm going to take, say, a series of 30 photos, I usually end up taking maybe 4 a minute, something like that. So if I take 100 images for a focus stack, 25 minutes right there.
Yeah. And this is obviously a well-dialed process at this point, because just you describing that process of twisting that focus ring a little bit, we're talking about, much less than millimeter increments of focusing. I would imagine that takes some practice. I could see myself ham-fistedly twisting that ring and just throwing it all off.
I mean,
Right.
I can actually see in my early photos, if you don't make the steps small enough, you actually see bands of blur in your subject. And
Uh,
I have one photograph that won a competition a long time ago, and it was hanging in a museum for a while. And I still cringe because I can see little bands of blurriness in it. It was one
yeah.
of my early attempts. And I want to say one other thing. I'm not always using the same camera settings. Aperture settings are different. So if I change the aperture, that also changes the depth of field, and that changes how much I need to rotate the focus ring each time. So having done this now for 2 or so years, it's a little bit of muscle memory. So I'm pretty used to what I need to do for different aperture settings. It all amounts to a single photo that I take in the field. We're not talking about work at the computer at home, but just in the field, a single photo will easily take me half an hour. Most commonly, it's between half an hour and a half to take what's going to end up being one photo.
Yeah.
Might be an hour and a half of work. Then I come home and I do a lot of processing of the photos to make sure everything looks good. And I use the focus stacking software. When you use the focus stacking software, it creates artifacts, which I'll not explain here, but that requires touch-up work, which I usually
Mm.
do in Photoshop. So any photo that I've done, actually either in the field or at home, can take me easily 5 hours of work. There's been some photos I've done that have taken me 20 hours just to come up with that photo. There was one that drove me crazy, that I was using the microscope lens at home and I just couldn't get the lighting and everything exactly the way I wanted to because the adjustments are so ridiculously tiny when you're looking at the microscopic level. I think I spent 40 hours creating one photo.
That's inconceivable. And is it for the microscopic work at home? Is that similar with this focus stacking idea, but just on a even smaller level?
It is similar in concept, yes, but when you're working at that tiny a level, with a microscope lens in particular, the depth of field is 7 microns. That's
Oh
seven
my!
thousandths of a millimeter, okay? 7 thousandths of a millimeter. That's the depth of field.
No
So
hand is
obviously,
twisting that. Yeah.
no hand is twisting that, and there's no focus ring on the microscope lens anyway. So at home, I use something called a focus stacking rail.
Mm.
It is a rail with a screw motor drive, and there's an electronic controller, and you program the electronic controller so it moves the camera along this rail in tiny, tiny steps. And the device that I have is capable of moving the camera at 1 micron steps, 1 micron. So when I use the microscope in the field, as I said, I take typically between maybe 20 and 100 photos. When I use the microscope lens at home, I'm taking usually between 300 and 500 photos to composite into one resulting photo.
That is absolutely incredible. I mean, what is the smallest organism that you've ever photographed? Now we're in the world of microns. So what is the smallest organism you've ever
Yeah.
captured?
So actually, the smallest organism I've ever captured is not a myxomycete. Two that I can think of. One is there are lots of tiny, tiny ascos. So I went into the field with a macro lens with the idea of taking photos of myxos. But when you're looking for tiny, tiny myxos, you find tiny, tiny ascos.
Right?
So then I started taking photos of tiny, tiny ascos. And people ask me a lot about mushroom ID, and I don't know that much about big mushrooms, but I know a lot about little mushrooms, tiny
Right.
mushrooms. So I found tiny ones. And my favorite one is one I still call the sunflower mushroom. When I saw it in the woods, I saw just a dot of yellow color, literally a dot, which I should say is what I'm looking for when I go in the woods. I just look for dots of color.
Yeah.
And then I look more closely with a magnifying lens. So I saw a yellow, sort of a lime, almost a lime yellow dot of color. And even with the magnifying lens, I just couldn't make out what it was. In that case, I took it home and I photographed it with what I had at the time. My microscope lens is 10x. At the time, I had a 5x lens. So I photographed it with a 5x lens, and they look like little tiny sunflowers. It's amazing to me the amount of detail at such a tiny level.
Right.
So that one is Neodecisifa serena. And those fruiting bodies are between maybe 0.2 and 0.5 millimeters. And they're just astonishing amount of detail at that level. It still blows me away when I look at them. The other one, which is actually my favorite all-time photograph, favorite thing I've ever found in the woods, again, it was a dot of color. Inside a crevice in a rotten log, I saw something that was bright orange. I looked with the magnifying lens, and I could actually see something almost looked like really, really, really tiny, tiny slime molds. But I'd never seen slime molds that tiny. So I took it home and I photographed it with, at the time, again, I just had the 5X lens, and they looked like Dr. Seuss trees. They're just these gorgeous, bright orange, like lollipop trees. They're just amazing. And I didn't think they were Myxos. I think I put them on the California Mushroom ID Facebook page. And even though I knew they weren't mushrooms, just to see if anybody knew what they were. And somebody actually came back within half an hour and told me it was probably Myxobacteria. So these are bacteria that behave a bit like slime molds in that they swarm together and they glide along. Myxo means glide, and they glide along the substrate in search of food. And when food becomes scarce, just like slime molds, they merge together and they create fruiting bodies. So that's actually the coolest thing I've ever photographed, and the smallest I've ever photographed. I think they're amazing. And I've managed to grow some in my moist chambers at home. So then I've photographed them with my microscope lens. I have several of those photographs on my Instagram account.
Immediately when you said lollipop Dr. Seuss forest, I thought of the image, and I was like, I'm pretty sure it's that one that I have in my head from your Instagram page, and they are stunning.
So I wanted to check that they were actually, I mean, somebody was just guessing. So I looked online and I couldn't find any photos of them online. And I searched long and hard. I found some drawings of them that looked like they were that. And I found some microscope images that sort of looked like that. So I just looked on the web to see if I could find somebody who studies these things. Right. And I found the name of a professor at UC Davis who studies Myxobacteria. And I thought, what the heck, I've got nothing to lose. And I sent him the photo and said, I found this in the woods. I think this is Myxobacteria. I see you studied them. Can you just take a minute and take a look at the picture and let me know if that's what they are? I got a response in literally 2 minutes. And the first word of my response was, wow, I've never seen them amazing. Like that. Yeah, so he confirmed that they were Myxobacteria of the genus Stigmatella. And he liked them so much, he actually put one of my photos on his lab's webpage. It's the featured photo on his page.
That's incredible. And I was going to ask how obviously you found a professor who's into Myxobacteria, but it's not something that everyone is fascinated with or even aware of. So, what kind of community have you found coalescing around these tiny organisms?
It's been great. And that's been actually a huge surprise to me that I have found a community. So I was taking photos just for fun, just for myself, just because I was fascinated by them.
Right.
And I had a friend who kept pushing me to post on Instagram, and I was resistant because I never did any social media until that point. And she kept nudging, we joked, she was kicking me. And I finally relented and I started posting on Instagram, and I thought I would get, maybe 100 followers would be exciting. And I got so excited when I hit 100 followers. Well, it definitely took off, that's for sure. So I started meeting, quote unquote, meeting online a few people that way. Leah Benlin, Leah Mycelia on Instagram was one of the first. And I actually wanted to learn a little bit more about mushrooms that I was photographing. So I started following people on Instagram, and Leah was one of the early ones. And I followed her because she was obviously very knowledgeable, and she was very kind in her comments and suggestions to people. But over time, I started gathering people who were more and more interested. What's been great for me on Instagram, my favorite comment on Instagram is when somebody says, wow, I never knew these things existed. You've opened my eyes up to a whole new world.
Right.
That is my absolute favorite comment. And that's my goal, is to show people these things that they might not have known ever existed, and to show people these fascinating tiny worlds that are right at their feet as they walk through the forest floor. So that's my favorite comment. When I first started, there was nobody else doing photographs like mine at a very, very detailed level. Not that I could find. There is a woman in Tasmania named Sarah Lloyd, and I want to add hers to the notes, who was the only other person when I started who was photographing Myxos and posting them online. She's very, very knowledgeable. She's been studying Myxos on her bit of land. She lives basically in a forest. And so she's been studying them for about 13 years and documenting them and studying them very carefully. So she was the person I actually learned the most from at the beginning. Then I started reading and getting a few more books. But over time, between Instagram and then I joined Facebook as well, because there is a slime mold appreciation and identification group on Facebook, which I also want to put a plug in for because it's great. Yeah. It's grown tremendously since I first joined. I mean, slime molds have become a little bit more well-known now for a number of reasons.
They have, yeah.
Yeah. The Paris Zoo had a little slime mold exhibit. Nova did a special on the intelligence of slime molds about 2 weeks ago, which is fascinating. I think they're just becoming more known. When I first started, there was only myself and Sarah and Leah occasionally who would post photos of slime molds, but I was doing slime molds more than anything else, and Sarah's doing slime molds only. I know that I've inspired a number of photographers because they tell me and they ask questions. I've helped a lot of photographers. If people ask me questions nicely, I'm happy to give advice. So
Yeah.
I've helped a lot of photographers with what gear to get and how to find them, how to photograph them, all those kinds of things. So now there are a fair number of very good photographers doing slime mold photography, almost focusing, no pun intended, on slime mold photography. And I think they're becoming more common. And on Facebook, which I definitely want to recommend that group consists of anything from rank amateurs, people who post a photo with their cell phone and say, did I find my first slime mold, to world-class experts.
Wow.
And everybody's very friendly. The world-class experts are really helpful to people. There are a lot of knowledgeable people who will answer questions. It's a really good way to see a variety of slime molds from around the world and learn from experts.
Yourself, Sarah Lloyd there in Tasmania, both opened my eyes up, and obviously Leah Mycelia, both opened my eyes up to this world of slime molds. And now I'm seeing it on Instagram where it's this, slime mold Sunday. It's this whole hashtag, and there's a whole movement around it to appreciate these tiny organisms. And I think you have played no small part in really pushing that trend forward. And it sounds like the online community and meeting people and just sharing this appreciation for such a tiny organism has really enriched your experience of photographing them. I mean, obviously there was an inborn drive to do so. It was a hobby that you were cultivating, but it sounds like that's really deepened the experience with you, has been able to connect with community and give back like that.
Completely. And even more so, more than saying it's enriched my photography, I would literally say it's enriched my life.
Yeah.
That was one morning, 3-4 weeks ago, when I woke up and I had messages from people in Russia, England, Sarah in Tasmania, Germany, and Norway, people that I communicate with on a regular basis. So especially during the pandemic, when we're largely staying at home, the photography has been a great distraction. So I've really upped my game this summer. So that's been a really good time to learn, but also being able to interact on a daily basis with so many people. This community of Myxo lovers has been just terrific. I mean, I have a great network of friends, which I'm very, very grateful for, but having this other bit in addition has been incredibly enriching, especially during the pandemic.
I mean, this is social media at its best. We all know about the failings of social media, but this is like what it was meant for. And I'm curious, did you have any innate fascination with biology before in what I want to call your first career, because this is your second career? Did you have any fascination for biology, or did Myxos kind of open your world up to kind of the study of biology?
I would definitely say I did not have a fascination with biology. I'm actually quite squeamish when it comes to a lot of biological things. But I've always loved nature. When I was a kid, I grew up outside New York City, and my parents' explorations were all of the cultural kind. So I never went or did anything in nature as a kid, except I always loved storms, and I would sit and just watch the rain and storms. I've always loved, and I love just sitting at the window and watching the rain, and I could do that for hours. Yeah. So I've always felt really good with rain. I love being in the rain. And when I went to college, again, it was all cultural stuff. And it was only when I went to grad school and I started running pretty seriously. I lived right next to the University Arboretum in Madison, Wisconsin. And I ran through all of the trails in the Arboretum and really started noticing all the changes of the seasons in there, pretty strong changes of seasons in Wisconsin. And it was really gorgeous. And then my graduate school boyfriend was from Minnesota, and his parents had a cabin in the woods in Minnesota. And that was my first time really being in the woods. And I just thought they were beautiful. And then coming to the Bay Area, the redwoods, they're just spectacular. So my favorite place in the world is a wet redwood forest. Really, really, really beautiful.
I don't think you can get much better than that. And that's really interesting to hear how your relationship with nature developed. And now you're so familiar with the smallest inhabitants. As much as this has given to you and enriched your life, what was it like to start getting into publications? Because now your photographs have stunned so many people. I guess, what was the first publication that reached out to you? What was that like?
Well, there's a funny story about this one. The first publication was a website called Colossal. This is Colossal, which I've seen for years. It's a very sort of artsy visual arts website. And I was having dinner with my friend Julia, and at dinner one night, somehow I got to talking about mushroom photography. My friends will laugh about that because
Of
I always
course.
talk about mushroom photography. So I
Right.
was talking about it, and I was telling her about a guy named Steve Axford, who's from Australia. I think you've interviewed him. He and Taylor Lockwood were my first inspirations for really good mushroom photography. And also, I saw some of Steve's slime molds photographs that first night. Anyway, I was having dinner with Julia, and I was telling her about Steve and how he was just like me. He was retired. He bought a camera. He started taking pictures in the local forest for fun. And Colossal picked him up and wrote a story on him. And all of a sudden, he became very well known. And I said to Julia, that would be really cool to be in Colossal. Yeah, right. Like that's ever going to happen. And literally
No
that very
way.
night, I came home at 10 o'clock at night. A friend of mine sent me a message that said, I don't want to call you. I'm not sure I'm awake. But did you see your photographs are in Colossal?
Literally, the universe picked up on what you were putting
It
out
was
there. And
incredible. I mean, I was thrilled to be in Colossal, but I was more than that. I was just astounded that I just said at dinner, hey, that would be great to be. And there it was.
more than
So,
coincidence.
so Colossal generated a huge amount of interest, and my followers grew quite dramatically within the next few days. And then Board Panda and a whole bunch of other websites picked it up. So I've been in a
Right.
whole bunch of web publications since then, and then various magazines, print publications. The best was, for me, the most exciting was the New York Times Sunday magazine, which is another one I said, I'd love to be in the New York Times someday because
Right.
I'm from the New York area. The New York Times is my home paper. I still read it every day. And yes, it happened. So they had an article on slime molds from a guy who's on the peninsula, and he told them to use my photos for the article. So there's a nice collage. And to see that almost a full page in the New York Times Sunday magazine was very exciting for me.
I mean, that's
Yeah.
incredible. Myxos, in a lot of ways, made some dreams come true.
Yeah. Which I didn't know
Being,
I had.
yeah, dreams you didn't know you had, but being in the New York Times like that, can't imagine how fulfilling that must be to go out in the woods and do this kind of work that you're doing, where it's so precise, so painstaking, to then get that kind of gratification to create this community and have people recognize it. That's just awesome. That makes me really happy for you. And it's just exciting to see these organisms garner so much interest and the people
It
who
is.
showcase them.
And when I first started doing this, it never, ever, ever occurred to me that any of this would happen. I'm just really enthusiastic about what I do, and people tell me my enthusiasm is contagious.
Yeah.
So I'm pretty enthusiastic in my postings. I get excited in both Instagram and Facebook postings. They're just fascinating little organisms.
That enthusiasm is always what seems to drive things forward and what people really resonate with. I'm curious how your husband's adapted to living with a Myxos celebrity superstar.
He's my number one fan for sure. He's very supportive. We hike together a lot. We're ardent hikers, so we still hike together a lot. I
Yeah.
never take a real camera with me into the woods when I'm hiking with him. I just have my cell phone with me. But when I do the photography in the woods, I'm almost always by myself because, as I said, it takes me an hour to do one photograph. People aren't going to wait for that.
Right.
But he's very supportive. And I try to mostly do the photography, especially the microscope photography when he's not at home, because it's so sensitive to any vibration. But if he's there, then I tell him, okay, you have to sit and not move. And I tell him when I start shooting and when I stop. So and during that period, don't move.
I figured he must now be obsessed with Myxos, but I love to hear that he's your number one fan. And this is kind of a more general question, but what has such a close relationship with these tiny organisms? What have they taught you? Because so many people talk about that when they get into biology. Whether you're studying flora, fauna, or fungi or microorganisms, what have they taught you in your time communing with them?
So the first thing that came to my mind is just how awesome nature is. I mean, I'm really a big nature fan. I love being outdoors. I love seeing wild lands, large, huge landscapes down to little tiny, tiny landscapes.
Right.
But I never really realized how awesome nature was at this tiny, tiny scale until I started photographing these things. It's just astonishing to me. I really feel like a little kid when I'm in the forest. I just get excited. It's
Yeah.
it's so exciting to me.
nature, even at its most grand, inspires awe, and inspires just as much awe at its most microscopic. Really beautiful. And then what's in the future for you? I mean, do you have any projects, plans, exhibitions coming up?
I have had a couple of exhibits. I had an exhibit at the first thing I agreed to was at my local library, which actually opened up a lot of doors, which was fun. And I've had a couple of exhibits since then. There's another one coming up. It's postponed indefinitely. The Exploratorium, which is the science museum in San Francisco,
Yeah,
has
tremendous.
a series every Thursday night for adults only. So it's adults only because they serve alcohol. And they have a topic every week. Now they've postponed those obviously since March, but there was one night that they had on fungi, and they asked me, and I agreed to do a display of the photos. And we actually had set up a very large screen in the middle of the main hallway in the Exploratorium, and I was just going to have a slideshow and talk about the Myxos and the tiny fungi as people walk by and come up and look and have questions. So that got postponed, but it will happen
Yeah.
whenever the Exploratorium opens. I assume, and they've said, that they will do that show again. And that was really fun to see the photos on a very big screen. A lot of people have asked me about a book, and at some point I'd like to do that. I think it would be fun to do that, but that's a ways off. It's not, I mean, it would be fun to do, but it will also take a lot of work. But mostly I just want to keep learning about them. I'm probably going to get at some point an even more magnifying. Instead of 10x, I might get a 20x microscope lens just so I can see even more. I don't actually have a microscope, which I probably will get at some point, because if I really want to ID the slime molds, you can usually ID to genus just from the photographic features. But if you want to get the species, you need to look at the spores pretty typically,
Right.
or other features. So I may get a microscope just for fun. Also, when you're growing Myxos, to find them with just my little tiny hand lens is pretty hard. It just takes a lot of time and patience. But if you put them under a microscope, it's a lot easier to find them. So
Right.
that's another possibility.
And then where can people find you? And can we really, what I want to know is, can we get giant prints of your work to adorn our homes and our offices?
Yeah, my favorite giant print had a little lineup, photograph I took in Alaska last year, a little lineup of a beautiful species called the Dymium squamulosum on a beautiful piece of rotting wood. And somebody, in fact, who works at the Exploratorium wanted to take that print and blow it up into a huge vinyl banner to put on in his backyard to hide something unsightly. So that's the biggest I've done. But I do sell prints. It's not my goal, but if people want them, yes, I'm happy to. I do sell prints, and basically just contact me on Instagram or Facebook. So I don't print them at home. I work with a lab, a very good lab. So they can buy prints from me online, and they can find me, of course, on Facebook and Instagram, or contact me on Facebook and Instagram.
And I know on Instagram it's Marin Mushrooms. And
Especially
then,
Marin_mushrooms.
thank you, Marin underscore
Yeah.
mushrooms. And then on Facebook, is it just your name, Alison Pollack?
It's with the middle initial K. So it's Alison K. Pollack, and Alison has one L.
We'll put the link in there so people can reach you. And I do encourage everybody to follow you, because if you're into fungi, if you're into really any kind of biology, and there's few things more stunning than these blown up pictures of these tiny, tiny organisms. And it's just incredible at even that scale, the amount of detail, the amount of vibrance. It's really beautiful. And you're providing us a really unique lens into that world. So I
Thank you.
personally appreciate your work and encourage everyone to go follow you. And I've actually adapted my last three questions that I asked all my guests a little bit, because obviously we're in the world of Myxos, not the world of mushrooms. So I think you've hinted at a couple that you really like, but what is just another Myxo, another slime mold, I guess we could say another Asco too, that you love, and why, that you want to share with us?
I'll give you two Myxos. One is called, at least the way I call it, is Diachia leucopodia. And it has a common name. Most Myxos do not have common names, but a few of them do. And this one does. It's called white-footed slime. It starts out white, as many slime molds do, and then the fruiting body is dark, it looks black. And then they change, as it dries out, they change to these iridescent bands of purple, blue, brown, just stunningly. You can't see this with the naked eye. It's even hard to see with the magnifying glass, but with a good magnification lens, either 5x or 10x, you can see this amazing iridescence. Somebody gave me a sample of it once that they grew in their garden, but I've never actually found it in the wild. And it does exist. People have found it a couple of times in the Bay Area. You can see that on iNaturalist. So that's one. And the other is a genus called Lamproderma. And I have found Lamproderma a couple of times, but only a very small number of them. But I love them also because of the iridescence. On my Instagram page, I have 2 fruiting bodies where you can really see the iridescence of these. They are just gorgeous. Blue and purple iridescence. They're stunningly beautiful.
And I know you started out saying the caveat you're not a scientist, but I must say you have a very good working familiarity with the scientific names and are incredibly knowledgeable. I've learned a lot about this microscopic world just from having this conversation, and I knew you'd have some great examples to share with us, so thank you. And then what has a relationship, and we've kind of talked about this and what they've taught you, but what has this relationship with Myxos, with these tiny organisms, given to you and really brought to your life?
2 things. One is, again, deepening my awe of nature, just to see. The first time I saw at the microscopic scale that Neodesicifera serena made large, it just amazed me that this little tiny 0.2, 0.3 millimeter fruiting body has all these hairs. And sometimes with the tiny ascos, they actually have crystals at the ends of the hairs, which you can see. Some Myxos have crystals, lime crystals on the surface, which you can also see if you magnify enough. So it's just really deepened my already good awe of nature, but it's really, really deepened it. And the other is to give me this, as I said, this community of people. Those discussions, whether they were online and then turned into chat or email, people have become friends through this photography. I think the photography for me has opened up a lot of doors to meeting people around the world. And I am a people person, so it's been great for me to meet people of different cultures all around the world.
And then the final one, and again, we've nibbled around the edges of this, but what is the lasting impact you hope to make with your work? Obviously, you didn't start out with some grand plan necessarily, but what's the lasting impact or what you hope people take away from interacting with your work?
I hope that people will become as much in awe of nature at this tiny scale as I am. So again, my favorite comment is, I never knew these things existed. So the more of that I see, the happier I am. And when people say, this is just amazing, I never knew it was so detailed and so beautiful in this little dot. I want people to be in awe of nature, and I want people to stop in the forest. I love it when people tell me, you've gotten me to slow down in my hikes. I will put in your show notes a link to this LED lit magnifying glass that I use, which is, I don't know, maybe $10-15, very inexpensive, but it's the best thing that you can use to look for Myxos.
That's fantastic. Thank you, because I think everyone now wants this magnifying glass to go out and find Myxos. And what a tremendous thing to be able to share as humans. That's one of the greatest gifts we have, is the awe that we feel in natural settings. The fact that you're able to share that to people is one of the greatest gifts you could possibly give. So, Alison, thank you so much for your time and really being open and very enthusiastic and passionate and really teaching us all, myself very much included, about this very small world of Myxomycetes. I'm fascinated and going to continue my own deep dive into this, but thank you so much for your time and coming on the Mushroom Hour. It's been a pleasure.
You're most welcome. It's been fun.